Thank you all. As Joe said, the session is going to be recorded, and I will be pausing along the way to give people time to ask questions, and also there'll be questions at the end. If you do have anything and you don't want to be recorded, or you don't want to interrupt, just type away in the chat function, and Joe's keeping a bit of an eye on this. So as I say, welcome. It's so good to have so many people on this Teams meeting. So this is extremely basic microbiology, and it really is quite basic. There will be some people on this call who already know quite a bit about microbiology. There will be some people on this who know nothing at all, and that's absolutely fine. And really the aims and objectives of this session are to have a little bit of an understanding about what is microbiology. We're going to be concentrating on bacteria and viruses, but there's far more to it than that, but that's what we're going to be focusing on. We're going to look at the chain of infection, and you're going to have a bit of an appreciation about microbiological testing and its limitations. And as I say, we are not going to go into any one infection in any detail. This is an overview. And it's also important to say that this is quite complicated stuff, and we've got an hour to cover microbiology, and if you become a microbiology doctor, it's a 5-year training course. So this is really a very basic overview into some quite complicated things. But there is, for those of you who are interested, and for those of you who want to find out more, there is further information in the links and in the further reading. So for those of you who want to go into more detail, absolutely please do. That would be really good. The kind of topics that we're going to cover, we're going to look at what the different types of microorganisms are, and really we're focusing on bacteria and viruses. I'm not really going to discuss parasites or fungi, although they are types of microorganisms, but for the purposes of this session, it's bacteria and viruses that we're going to be looking at. We're going to look a little bit about disease transmission. We're going to look at microbiological testing, very basically. A bit about the chain of infection. Actually, I've just realised that we're not covered incubation period or period of infectivity, but that's not a problem. That's going to be covered on a later session. But we're going to be also looking at multi-drug resistant bacteria. And as I say, we're very much concentrating on bacteria and viruses. And I do want to give a bit of a warning here. I am not a microbiologist, and as I say, this is a very brief intro session. So for those of you who want to, please do read into this further, and there's some really good online training, there's some really good websites that I've linked to that you can get a much more detailed appreciation of this topic. So with all those caveats out of the way, what is microbiology? So microbiology is the study of microscopic organisms and how they interact with humans and the environment. And by microscopic, we mean that they're small. They are too small to be seen by the naked eye. You have to use a microscope to see them. And microbiology is just any life that is too small to be seen, as I say, by the naked eye. And there are, these things are tiny. I'm going to show you a bit more detail shortly, but basically hundreds of thousands of bacteria can fit onto that full stop there at the end of this sentence. These things are absolutely tiny. And there are 4 main parts of microbiology: bacteria, viruses, fungi, and parasites. We are, as I say, concentrating on bacteria and viruses here. I just wanted to elaborate on that slightly further, because like anything else, it's actually not that simple. So this is a diagram of life. So all life on Earth fits into this tree. This is the classification of life. And you've got the bacteria over here. So bacteria are very simple, single-celled organisms, and they're over here on this side. You've got the archaea, which again are, they're also very simple. They're single-celled organisms. They're different to bacteria. They evolved separately, but they are life and they are very, very simple. So here and here, we've got very simple single-celled life forms. This middle part of the tree here, it's much more complicated. These are the eukaryotes. So these are multi-celled organisms, and this includes everything else. So we've got the plants up here, we've got trees, we've got all plant life is here. We've got fungi, we've got parasites. These are quite complicated organisms. They're not single-celled like the archaea or the bacteria. And then we've got animals. And obviously animals includes the mammals, of which we are one as humans. We've got snakes, lizards, turtles, fish, bony fish, insects. So all of life fits into this classification. And bacteria are over here, very simple, but they are living organisms, as are fungi. However, I've put viruses and prions over to one side over here because these are not living organisms. Viruses are not alive. So bacteria are alive. They can survive outside of, they can survive on their own, they can multiply. Oh, sorry about that. They are living organisms. Viruses are not. They are not alive in any sense of our understanding of the term. Viruses don't live. Also prions. Prions aren't living organisms either, but they can cause disease. And some of you, I'm sure, will have heard of mad cow disease, which is caused, which is an infection caused by new variant CJD. And it's a prion, and prions are just a fold of protein. They're just a scrap of protein. They're not living, they're not alive. They're not alive in any way that we understand the term, but they can cause disease in humans and in animals. And viruses are the same. They're not living. So when we talk about killing the virus, we don't mean we're literally killing it. You can't kill something that's not alive, but you can deactivate. So this is really just a bit of an overview of life in general, just to show you where the bacteria and the viruses fit into this. And one of the things I wanted to just show is how small these organisms are. So when we talk about microbiology, we're looking at things that really can't be seen with the naked eye. We're all familiar with things like chicken eggs, ostrich eggs, perhaps so much so, you know, adults. We look on, we see things on this sort of scale. And really, we can obviously see each other. We can see a chicken egg. We can see a frog's egg. We can see frog spawn in the pond. All of these things are visible to the naked eye. Really, when you're getting down to the size of a human egg, these things are really just on the very borderline of things that we can see with our naked eye unassisted. And then we're looking at our cells, so animal cells, plant cells, we're all made up of cells. We can't see these. These are invisible. We can't see these with our naked eye, and nor can we see the bacteria and the viruses and the proteins and these infectious organisms that causes infection, and right down to the atom, which is the smallest thing that there is. If you split the atom, you get nuclear fission. So really, this is just a scale from the things that we can see with our naked eye down to the things that we cannot see with the naked eye. Bacteria we can see with an ordinary microscope. Viruses we can't. We have to use a different type of microscope called an electron microscope. And I just want to make you, give you some concept of the scale. So we all know about kilometers and centimeters and millimeters, and of course there are 10 millimeters in a centimeter. But the next level down from millimeters, which we're all familiar with here, is the micron, the micrometer, and there are 1,000 micrometers in a millimeter. So if you just think how small a millimeter is, you can see a millimeter here on the ruler, 1, 2, 3, 4, 5 centimeters. Sorry, can people still see this presentation? So if somebody else is sharing, can whoever that is please stop?
No, can't see the presentation, Valerie. Can't see you though.
Somebody else is sharing this presentation. Can whoever that, or sharing their screen, can whoever it is please stop doing that? I'm going to try and go back. Can people see now?
Yeah, it's back on.
What screen are you looking at?
Bacteria and virus.
Okay, I'm now seeing something completely different to all of you, so this is going to be very interesting. So do bear with me, and sorry about the little hiccups there.
We are back on the diagram now.
Brilliant. So we've got that very small scale. So my pointer is working. So microns. There's 1,000 microns per millimeter. So these things are absolutely tiny. So it's just to give you some idea about the scale of these things. They really are little. And as I say, you can fit thousands of bacteria on a single full stop at the end of a sentence. So that's just to kind of give you a bit of a sense of the scale and where all of this fits in. So we're now going to talk about bacteria and viruses in a little bit more detail. And as I say, this is a very brief overview of the subject. So what are bacteria? I'm just going to, sorry, I'm sorry about that. There's a few IT glitches today. So bacteria are alive. They are living organisms. They are single cells, so they're not complicated. They're not like plants or humans or mammals or fish. They are very simple organisms, but they are living, and they are single-celled organisms. The cell doesn't have a nucleus, so all of the things that makes the cell work, and sorry, my pointer isn't, oh, it is, yeah. All of the things that make the cell work are either DNA or RNA that is in the middle of the bacterial cell there. The bacteria is surrounded by a capsule and a cell wall to keep it all in and enclosed, and there might be a membrane as well. And you often have these little flagellum, these little wiggly things here, and these help the bacteria swim around, basically, and help them transport themselves from place to place. So this is really what bacteria, this is a simplified diagram of what bacteria look like. They're single-celled, they've got this DNA oil in the middle of the bacteria, and they reproduce by binary fission. So they, unlike humans or mammals or animals that reproduce by sexual contact, so you need a male and a female, a sperm and an egg, bacteria don't do that. They reproduce by binary fission. And we're not moving forward.
Valerie, are you going to explain what binary fission is?
I am. I'm trying to move on to the next slide and I can't do it. I'm going to try and I'm going to start the show again because the slides that I'm seeing are completely different to the slides that you guys are. So let me try this. Right. Can you see the what are bacteria slides?
Yes.
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 We're back on track. So sorry about this. The IT gremlins are really striking today. So bacterial reproduction, so binary fission. So bacteria reproduce in this way. So basically you get a bacterial cell here, and that we call that the parent cell. And basically the DNA that's in the middle of the bacteria there, it creates a duplicate of itself. So this is the DNA that we saw at the middle of the cell here, and it duplicates itself. So we've now got 2 DNA duplicates. And then the cytoplasm, the sort of the bit around the bacteria, it divides. And now we've got 2 daughter cells. So from one original parent cell, we've got 2 identical copies daughter cells. So this is how bacteria, this is the main way that bacteria reproduce, by binary fission. So unlike with, humans, we have a sperm and an egg, and the child receives genetic material from the mother and the father. This is asexual, and it's these daughter cells are a clone of the original parent cell. They're exactly the same. And again, you can see how quickly it can happen. So 1 cell here becomes 2, 2 cells then become 4, 4 cells will then become 8, 8 will become 16, 16, 32, 64, 128, etc., etc. So you can see that very quickly one single bacterium can very quickly become thousands and thousands and thousands of bacterial cells that cause infections. Now, when I say that the daughter cells are clones, they are, but little changes can happen, and they can become slightly different over time, and I'll come on to that later on when we talk about how antibiotic resistance. So this is just a simplification of how bacteria reproduce, and there's more information on these links at the bottom of the page there, but really it's just to show you that a bacterium can divide on its own. It doesn't need another bacterium to do so, and that because you've got this exponential growth, it can happen very, very quickly. So very quickly you can go from one bacterium to an entire colony of bacteria that can cause infections. I did want to talk to you about how bacteria are classified, because this is talked about a lot in microbiology, and it can be a little bit bewildering for people who aren't quite sure what the jargon means. And it's fairly basic in its concept. So we classify bacteria depending on their shape. So some of them are round, and we call those the cocci. And you can get diplococci, which is just 2 bacteria, 2 of them stuck together. And Streptococcus pneumoniae is an example. You can get staphylococci. So again, this is Staph aureus that you've probably all heard of, and it's just, this is the shape of the bacterium. You can get streptococci, which is kind of a long little wiggly worm shape, or you can get 4 together or several more together. So you can see some of them are just round shapes, and we call the ones that are cocci, we call the ones that are round, we call them cocci. Some of them are more of a rod shape. So we call those the bacilli, and so you've got anthrax here, that is a rod. Salmonella typhi, again, is a rod shape, and you can see all these little flagella floating out from the typhi, and Clostridium botulinum as well is a rod shape. And then we've got the other ones that don't really fit into the cocci or the bacilli. So, what they look like is actually a really good way of classifying the bacteria. And you can see that you can get them in pairs, chains, or clusters together. So this is what they look like, and you can see these under the microscope, and this is how they look like. And again, there's more information on the link below. So another way of classifying bacteria is by gram staining, and some of you may have heard about this. I'm not going to go into any real detail, but I just wanted to make sure that you're aware of the concept. So most bacteria, and it is not all bacteria, so the mycobacteria, things like TB, the bacteria that causes tuberculosis, they don't fit into that. But most bacteria you can say it's either gram positive or gram negative. And it's really to do with how the cell wall is structured. And this is just a diagram of the cell wall of a gram negative bacteria and a gram positive bacteria. And I'm not going to go into any detail, but you can see here that there are differences in the cell wall structure between gram-negative and gram-positive organisms. And what happens is, if you we can do something called a gram stain test, and this was identified by a guy called Hans Christian Gram. And he realized that if you stain the bacteria plates with dye, they will look different depending if they are gram-positive or gram-negative. And when you do this staining, when you add the dye, the gram-positive bacteria are a blue colour and the gram-negative bacteria are this kind of red colour here. So that's just a very basic way of classifying bacteria. Most bacteria are either gram-negative or gram-positive, depending on the structure of their cell wall. And then I just wanted to talk about two common bacterial pathogens. So we've got MRSA. I'm sure you've all heard of MRSA. So this is on the left here. So MRSA is a Staphylococcus aureus, which is resistant to an antibiotic called methicillin, or a group of antibiotics, I should say. And it's a gram-positive cocci. So it's going to go blue under the when you dye it, and it's a cocci, so it's these round shapes here. And MRSA can cause wound infections, it can cause blood infections, it can cause septicemia, and as I say, it's multi-drug resistant, so it can be quite difficult to treat MRSA infections. And this is a big problem in hospitals, and lots of time and effort and money has been spent to reduce and fight MRSA infections by various means. So MRSA Staph aureus, it's a gram-positive cocci. So on the right here, we can see a very different shaped bacteria. These ones are small and round, these ones are rod shapes, and this is E. coli. So this is a gram-negative rod. It's a rod shape, and it's a kind of red when you stain it on the gram staining. And E. coli is a bacteria that lives in our gut. We've all got it probably in our bowels, in our guts. And it's fine in the gut. It actually makes vitamin K and it keeps us healthy and helps digest food. But if it gets out of the gut and into a wound, it can cause wound infections. And it can also cause UTIs, urine infections, and that's particularly common in women. If the E. coli gets out of the gut and into the urethra, it can cause urine infections, kidney infections, bladder infections. And again, lots of E. coli are multi-drug resistant, which makes them very hard to treat, and that's a real problem. And I'm going to touch on multi-drug resistance in a little while. Any questions so far?
Nothing on the chat, Valerie.
Good, and hopefully our gremlins have passed. So treating bacterial infections, we treat bacterial infections, as you know, with antibiotics. And antibiotics, we can take them as tablets or as pills, and we can take them orally. They can be topical, so it can be a cream or an eye drop or an ear drop. So topically just means if you've got a skin infection, you apply the cream directly to the site of the infection. Or we can give them intravenously. So somebody who's very unwell with an infection may require antibiotics injected directly into the bloodstream, so intravenous antibiotics. So that's really the three main routes of giving antibiotics. And antibiotics, they either kill the bacteria or they prevent the bacterial growth. So earlier on we looked at how they reproduce, and you can see that really quickly one single bacterium can very quickly become thousands and thousands of bacteria, which can cause infection. So an antibiotic that stops the bacteria from growing, from dividing, can be very effective. People talk about strong antibiotics. So I had an infection and I took antibiotics and it didn't work, so I went back to the doctor and got some stronger ones. And that's really quite a misleading term, we don't think about antibiotics as being strong. That's not how they work. Instead, we've got broad spectrum antibiotics and narrow spectrum antibiotics. Now, some antibiotics will work against a really wide range of infections, lots of different bacteria. That antibiotic will kill lots of different types of bacteria. And they're really useful if you've got a chest infection, you go to your GP. Most of the time, the GP is not going to take a sputum specimen to find out what the bacteria is that's causing your infection. They will just give you a broad spectrum antibiotic that the GP knows will kill most of the bacteria that are likely to be causing your chest infection. So they're really useful in that sense. However, that can cause problems with antibiotic resistance, and I'm going to come on to that shortly. There are also antibiotics that we call narrow spectrum. Now, these don't kill lots of different types of bacteria, they just kill one or two. So it's no good if you go to your GP and the GP is not going to prescribe you a narrow spectrum antibiotic, because how does he or she know that the antibiotic they've prescribed is going to work against the bacteria that's causing your chest infection? But these narrow spectrum antibiotics, they're really useful in a hospital setting or when we have got microbiological confirmation of what the bacteria is that's causing the infection. So we don't use the term strong. It doesn't really mean much. We talk about broad spectrum antibiotics that kill lots of different bacteria and narrow spectrum antibiotics that will only kill a very small select group of bacteria. And we can give those via injection, by tablet, pill, or by topical treatment. I don't expect you to remember this, know this, or be aware of this, but I just wanted you to be aware that there are really 6 groups of antibiotics. Some of which are used far more than others. I'm sure you've all heard of penicillin. Many of us will have had penicillin or amoxicillin. Some of you will be allergic to penicillin and can't have it. But really, this is just an overview of the 6 groups of antibiotics, some of which we use far more than others. So penicillins, for example, we use them very commonly. So penicillin was the first antibiotic that was discovered in the 1940s by Alexander Fleming. And we still use the penicillins today. They are very good at treating quite a wide range of different infections. And some of them we don't use very often. So the aminoglycosides, these are really only given in hospitals, and they can cause quite serious side effects. And they're normally given by injections. So there's just a wide range of antibiotics there, and there's some more information on the NHS website about the different types. And you can see here that, broad spectrum antibiotics, the fluoroquines, are used to treat a wide range of infections. And I do apologise, that's my phone going in the background, but we'll just ignore it. So antibiotic resistance, this is a so just apologise for the phone. So antibiotic resistance is a massive public health threat at the moment. And we're obviously all focused very much on COVID-19, and we will continue to be so. But antibiotic resistance is a huge threat to health. And so antibiotic resistance is a huge threat to health, and it's quite a complicated concept, and different types of bacteria become resistant to bacteria in different ways. So I'm not going to go into huge detail. There is more information on the link at the bottom here. But basically what happens is if you've got a large number of bacteria, and some of those bacteria, these kind of orangey ones here, they might be resistant to antibiotics. So the person who's got the infection takes antibiotics, and the antibiotics kill all the blue bacteria. So it kills all the bacteria that are sensitive, but the ones that are resistant are left behind. So these 2 bacteriums here, they are resistant to that antibiotic, and they are not killed by the antibiotic. But now they haven't got any competition. So these resistant bacteria can now multiply by binary fission. They start to divide. And now we have got somebody who is colonized with these resistant bacteria to which the antibiotic doesn't work. And some bacteria can even transfer their drug resistance to other bacteria. So whatever it is that's made that bacterium resistant, it can pass that on to different bacteria, which causes even more problems. Now, this is a gross simplification. This really is. It's far, far more complicated than that. But I just wanted to give you a bit of an overview. And I'd argue that, you know, pandemics aside, this is probably one of the biggest threats to public health that there is. And last week I showed you the picture of Marianne Dashwood, who'd been for a walk in the rain and had got a throat infection and almost died. And the risk is that we could go back to the pre-antibiotic era where people have infections that we can no longer treat. So antibiotic resistance is a big problem. We've all got a role to play in that. So antibiotics don't work against viral infections. So we need to make sure that they're being prescribed appropriately. We need to make sure that antibiotics are not being used in agriculture and in foods. And really it's a big public health threat. And when I give talks to doctors, junior doctors in the hospital, I always say to them, you know, this is going to be a massive problem for you throughout your careers, and it certainly is for anybody who works in public health. So there's more information in the link there about this, but it's a huge challenge for us all, and it's not going away, and it's only going to get worse. So I painted quite a negative picture about bacteria, and that's understandable perhaps because I work in health protection and healthcare, and we tend to think of bacteria as bad. They cause disease, they cause infection, and some of them do, but actually the vast majority of bacteria don't. We couldn't survive without bacteria. We wouldn't exist. Bacteria are one of the first life forms on Earth. We are covered in bacteria. There's probably more bacteria in us and on us than there are human cells. They're in our guts and they help us digest food and they produce vitamins. We wouldn't have beer, wine and cheese without bacteria. So I think you know, that's a great service they provide. They keep us healthy. They keep our skin healthy. I'm sure a lot of you have heard about the gut microbiome and how important the bacteria that in our gut is for all of our health, not just digestion, but for things that we never considered before. So the more we learn about bacteria, the more important we realise they are. So actually, it's probably worth remembering that the vast majority of bacteria do not cause us harm and actually keep us healthy and keep us safe and you know, boost our immune system and generally are very valuable. So even if we could, and of course we can't, even if we could, we can't get rid of bacteria, and we certainly wouldn't want to because they are absolutely essential to life. So any questions about bacteria so far?
Nothing on the chat.
Fab. So viruses, I'm going to move away from bacteria onto viruses. Viruses are very different. So viruses are not living. So early on we showed the classification of life, and viruses and prions were off to the side. Viruses are not living organisms. They are an infectious agents, and they do and can cause infections. But they are parasites. They are obligate parasites. And what we mean by that is that they cannot survive, they cannot exist, so they can't replicate. They can only replicate inside a living cell. So outside of a cell, they can't reproduce, they can't carry on with any metabolic processes, they can't create or store energy, they can't really do anything at all outside of a human cell or a plant cell. So that we call them parasites because they can't survive on their own. And a virus it's got a similar structure. You've got an envelope that keeps it all together. In the middle, you've got either DNA or RNA, depending on the virus it is, and it's all held together by this envelope here. And this is what virus is. This is a kind of diagram of viruses. So again, incredibly complicated. I'm not going to go into this in any detail, but I wanted to show you that it's different to how bacteria reproduce. So you remember that you start off with one bacterium, it divides into two, the two divide into four, etc. And they do so by asexual reproduction and create clones of themselves. It's very different with viruses. So what happens with a virus? So this is our little virus here. The virus has to enter a cell. So this is, say, a human cell. It doesn't matter. It could be an animal or a plant cell. The virus has to enter that cell where a very complicated process happens, which I am not going to explain, but there is more information there for those of you who are interested. And the virus creates replicants, and they are then released out of the cell. They burst out of the cell, and they then go on to another cell where they enter the cell, reproduce, and are released. So this is how viruses reproduce. And as I say, they are obligate parasites. They have to do this within inside a cell. They cannot reproduce outside of cells. Viruses are also classified. So we classify bacteria depending on their shape and depending on whether they stain with dye when they are gram stained. Viruses are a little bit different. So the two main ways of classifying them is, do they have DNA or do they have RNA? And then you can see underneath there are various different ways of classifying viruses. Again, I'm not going to go into this into any more detail. For those of you who are interested, there's some useful links there. But really, here we can see the coronavirus. So a coronavirus is an enveloped positive RNA virus. So that's where the coronaviruses fit in. And obviously COVID-19 is caused by a coronavirus, the SARS-CoV-2 virus. Which brings me on to SARS-CoV-2. So it's an RNA virus. So the genetic makeup in the center of the viron is RNA. And it's one of the coronaviruses, and it can cause COVID-19. And this is what it looks like under the microscope there. So you've got the RNA in the middle, you've got this lipid envelope around the outside that keeps it all in together. And this is kind of what it looks like under the electron microscope. Now this envelope it's important, it keeps the virus together, but also this envelope is actually really easy to dismantle. All we need to do to break down this lipid envelope, this fatty envelope around the virion, is introduce soap. So soap will destroy this, and once the envelope has been destroyed, the RNA and everything just falls out and it no longer exists. It's been deactivated. So that's why soap and water and really basic measures like hand hygiene are so effective against at preventing the spread of COVID-19, because this envelope around the outside of the virus can be very easily dismantled and disrupted. So viral infections, you can't treat viral infections with antibiotics because antibiotics don't work against viral infections. But we do have some antiviral drugs, and I'm sure you've all heard of Tamiflu, for example, that is a drug that's used to treat influenza. But these antiviral drugs, they work in different ways, but really they stop the virus developing. And we don't really have any at the moment that are specific for COVID-19, though obviously there's a huge amount of research looking at this. But most of the antiviral drugs that we've got at the moment are for dealing with HIV, herpes, also hepatitis B and C, and flu as well. The problem is, and if you remember, the virus has to replicate inside a human cell. Now, if the virus is inside one of our cells, it's really difficult to get a drug that is going to attack the virus without attacking our cells. So that's what makes it slightly more difficult, I think, than drugs that treat bacterial infections, because they can target the bacteria that's in our body, that's in our blood or in a wound or wherever it may be, but the virus is inside our own cells, and that makes it quite difficult to treat. And also viruses, they mutate and they change. So as I'm sure you all know, we have to have a flu vaccine every single year, because the flu that's circulating this winter will be different from the flu that was circulating last winter, and that's due to variation and mutation in the virus. And that makes it quite challenging in developing these antiviral drugs. So it is quite a bit harder to develop antiviral drugs than it is antibiotics to treat bacterial infections. Any questions about viruses so far?
Valerie.
Yeah.
There was a big talk about anti-malaria. I think I can't remember. It's chloroquine, something like that. You know, there was a big hype about it. Can you say one or two words about that, please?
There was a big hype, and it was interesting because I think it was a company that Donald Trump had shares in, and so he was promoting it and he was taking it. There was then a very interesting piece, I think it was the Lancet, I can't remember, that showed it was potentially harmful, and then it was retracted by the authors because of issues and discrepancies with the study. I don't know if Neil or Alice or Paul or Annie, if they're on the call, and if any of you know more about it than I do.
I read an article about the side effect of using it, and it was to do that it causes more heart attacks and other side effects. As I think that was the article in the Lancet, but I was just wondering in terms of how it works with the viruses, and considering malaria is a parasite.
Yeah.
So it was interesting, isn't it?
I don't know, and the other drug that's to answer your question, I don't really know, and I'm not sure quite where we are with that one. The other drug that's been in the media recently is dexamethasone, which is a steroid which reduces inflammation in the body. So like chloroquine, it's not an antiviral drug, it's not targeting the virus itself, but it reduces inflammation, and that's been shown to reduce mortality, so reduce deaths in the very sickest patients who are on ITU. So again, it's not a treatment, but it's something that it's a supportive therapy that can improve, prevent dying in the very sickest people. But at the moment, there is no antiviral, as far as I'm aware, there are no antiviral drugs that we know have any effectiveness against COVID-19. So moving on now, so that is viruses and bacteria in a very, very, very basic whistle-stop tour. And as I say, there's a lot more information for those of you who are interested, but we can only do a very brief overview in an hour. So I wanted to kind of bring this into the concept of the chain of infection, because you might be thinking, well, yeah, that's great. I didn't know much about microbiology before, but what does that mean for COVID-19? What does that mean for me working in health protection? And really, what we're trying to do with any infection, whether it's COVID-19, whether it's TB, whether it's cholera, whether it's malaria, we're trying to break this chain of infection. Now, in order for infection to occur, all of these things need to be in place. So if we can break this chain, we can prevent infections from occurring. So first of all, we need an infectious agent. That's the first part of the chain. Now, at the moment, we're talking about SARS-CoV-2, which is COVID-19, but it might be MRSA, it might be E. coli, it could be anything. But you need an infectious agent, whether it's a virus or a bacteria. This is what causes the infection in the first place. And that infectious agent needs a reservoir. It needs somewhere to survive. Now, it could be a animal reservoir. So COVID-19 we think is linked to bats. It could be chickens, it could be pigs. Lots of different strains of flu are bird flu or swine flu. They originally were diseases of animals, pigs or chicken or birds. The reservoir might be the environment. It might be in the soil. It might be in the water. The water that we drink is safe to drink, but it's not sterile. Humans, we might be the reservoir, or it could be in the food that we eat. But the infectious agent has got to be somewhere in order to cause infection. And then it's got to get out of somebody somehow. Now, many infections cause symptoms like vomiting, diarrhea, and that's the viruses or the bacteria's way of getting out of the host and getting into the next person. If you think about it, we shouldn't anthropomorphize, viruses aren't alive and bacteria don't think. But from the bacteria or the virus's point of view, what they want to do is get from one person to the next person. And a really good way of doing that is making the person vomit, have diarrhea. It's a really effective way from the pathogen's point of view to spread. It can get out through our blood, through our saliva, but it can get out of the host in various ways. And then you've got the modes of transmission. So different infections spread in different ways. First of all, on this side, you've got sort of general transmission. So these are things that happen in the environment. So the water, there might be Legionella bacteria in the water, for example, or there might be a mosquito that's carrying dengue or malaria, or a flea that's carrying bubonic plague. And then you've got the human-to-human transmission, and this is the bit that we're more interested in. Really, pretty much all infections spread in these 5 ways. So through contact. Now, contact can be direct contact. That could be sexual contact, that could be shaking hands, it could be hugging, but it's direct contact from person to person. And viruses such as HIV, Ebola, herpes can be spread this way. HIV obviously through sexual contact only or through sharing needles. And then you've got indirect contact. So if my hands are contaminated with a pathogen and then I touch that door handle there, the next person who comes along and touches the door handle can potentially catch the infection from me. And things like flu, norovirus do spread very well that way. Droplets. So droplets are heavy respiratory droplets that we all, as we're talking, we're singing, we're coughing, we're sneezing, we all release droplets into the air. And you can have pathogens in those droplets, and COVID-19 spreads very well that way. As we also talk and sing and cough and sneeze, we also release much smaller, finer respiratory secretions, and these stay floating around in the air for much longer. And these much smaller respiratory secretions can contain pathogens as well. So COVID-19 we do not think is spread by this airborne route. We know that infections like flu are, but we don't think COVID-19 is. We think it's mainly this droplet route. And then there's fecal oral. And really that's through contaminated food or water, or inadequate cleaning, or poor sanitation, or contaminated drinking water. So pretty much all infections in humans spread in these ways. And as far as COVID-19 is concerned, we're looking at droplets, and we're looking at direct and indirect contact. These are the routes of spread of COVID-19. And then it's got to get into us somehow. And pathogens can get into us in several ways, and really our skin is our biggest barrier against infection. But if we've got any cuts or breaks in the skin, then pathogens can get in that way. It can get in through our eyes. If we touch our eyes and we have contaminated hands, infections can come in that way. Infections like HIV and hepatitis are bloodborne and can be spread through sexual contact, as can things like syphilis and gonorrhea. The food that we eat and drink can be contaminated, and also we can inhale through our mouth and nose. And again, COVID-19, the way it gets into our body, it's a respiratory infection. It's really coming in this way. This is the main route of spread, although it could come through if we have contaminated hands and touch our eyes. And I put mouth and nose there together because you might be thinking, well, that's obvious, but the number of people I've seen wearing masks that just cover their mouth and not their nose, of course, it's doing no good whatsoever. So when we breathe, we've got to remember the nose as well as the mouth. And then in the chain of infection, we need a susceptible host. Some people are more susceptible than others, and we're still learning about that with COVID-19. But really, more generally speaking, the people who tend to be most vulnerable are the very young and the old, people whose immune system either hasn't developed fully yet or is getting worn out, basically. Anyone who's got a damaged or weakened immune system, so anybody who's, for example, is being treated for cancer with chemotherapy is more at risk of infection. Some infections we can vaccinate against, so measles and MMR. You know, if somebody has had the MMR vaccine, then they are not a susceptible host to measles because they are protected with the vaccine. We think there's genetic disposition to some diseases, and obviously a lot of work's going on at the moment with COVID-19, looking at Black and minority ethnic groups and why it appears that some groups are much more adversely affected than others. And that's quite likely to be very complicated and multifactorial, but there may well be genetic disposition there. But really, as far as infections and COVID-19 goes, any of us are potentially susceptible. Even young, fit, healthy people with no risk factors can catch and become severely ill or die with COVID-19, although the risk is far greater in certain groups. So just kind of rounding off the chain of infection there, we can't, we're going to look at this in more detail in future modules, but there's only some places that we can break this chain of infection. So we can't do anything about the infectious agents. Last week I said that smallpox is the only disease we've ever eradicated. We can't get rid of COVID-19 or SARS-CoV-2. We can't get rid of MRSA or cholera, sadly. These pathogens are here to stay. We can't do much about the reservoir. We can't get rid of people or the environment or water or plants. And we can't do anything about us. You know, we can perhaps vaccinate people and that reduces susceptible hosts. And we can do everything we can as a society to make sure everybody's as healthy as they can be. But really, these three areas, there's not a huge amount we can do about. But what we're trying to do is break the chain of infection here. And if we can stop the infection from spreading from one person to the next person by, for example, social distancing or cleaning our hands, then we can break that chain of infection. And we're going to look at this in more detail in future modules. So just for the last 5 minutes, and I'm running slightly late because of all our IT gremlins, I just wanted to talk to you very briefly about microbiological testing and how this works. With bacterial infections, it's actually quite easy, and a microbiologist even from 100 years ago would absolutely recognise this process. It's not really changed to any intents and purpose in 100 years. But what you do is, if you want to see if somebody's got a bacterial infection, you get a sample. So if it's a chest infection, you get a sample of sputum. If it's a urine infection, you get a sample of the urine. And you get that sample and you plate it up, basically. So this is a Petri dish. You plate up that sample on this Petri dish. And this is Staph aureus. You can see its little golden colonies of bacteria that's growing on this Petri dish. Now, this takes several days to do because you are literally growing the bacteria. You've got this plate, this Petri dish that's got nutrients in it, and you are growing the bacteria on the surface. And the bacteria are reproducing by binary fission, and it will take several days to grow. And then what you do is you'll take one of these little colony forming units and you'll have a look at it under the microscope and you'll say, aha, this is Staph aureus, or whatever it might be. So it takes several days. You don't get an answer straight away. So if I turn up to my GP with an infection, I'm not going to know what the bacteria is that's causing it for about 3 or 5 days. And also it's quite resource intensive. You need trained microbiologists to do this work, and you need trained microbiology doctors who can look at it under the microscope and diagnose on the basis for this. So that's how we diagnose bacterial infections. Viral infections are done in a different way, and I'm sure you've all heard of PCR because we're talking about it a lot at the moment with regards to COVID-19. But basically, got nothing to do, completely different from this. Somebody from 100 years ago would have no way of knowing what was going on here. Basically, what you do is you take a swab. So with COVID-19, it's a throat swab and it's a nose swab, and you get that swab really to the back of the throat to make somebody gag. And basically what you're doing is you're looking for the antigen. And in the case of COVID-19, you're looking for viral RNA. So an antigen, it's a foreign substance which creates an immune response in the body. And it's the antigen that our immune system recognises to produce antibodies. And Neil is doing a session on Thursday about our immune system and immunology, and he will explain it far better than I can, but there will be a bit more about that there. But basically this swab is taken, it gets put into a machine, and the machine will kick out a result really within a couple of hours, really very quickly. The longest bit of this process is getting the swab from the patient to the lab and into the machine. So bacterial infections can take several days to diagnose. Viral infections, if there's a way of doing this PCR testing, it can take a few hours. So there's a huge advantage there. You can get the results far quicker. But it's not foolproof. No test is 100% accurate, and that's just not possible. There is no such thing as a test that will always be accurate in all circumstances, and we can get false positives and false negatives with any kind of test. So a false positive is when somebody doesn't have the disease, but the test result comes back as positive. Now that's clearly not great because it will cause anxiety in that person. It may lead to unnecessary treatment. They may be given treatment that they don't need because, remember, they don't have the disease, but the test result has said that they do. So that's not a great circumstance. Probably even worse is the false negative. And that is when the person has the disease, but they get tested and the test result comes back as negative. And again, that's really not great. It's not great for that person because it means that they might not get the treatment that they need because they've got the disease. But also, it might mean there's a potential spread of disease. So, for example, if somebody had COVID-19, but they got a test and the test came back as negative, they might think, right, I don't need to stay at home. I don't need to quarantine for 7 days. I'm going to go down the pub. So they are potentially spreading the disease to other people. And there's a really good article there that this is a gross oversimplification of what's quite a complicated issue. But for people who are interested in reading more about it, there's a really good article there which talks about the positive and negative predictive value of any one test. So if you're interested, do have a look at that. So penultimate slide, just regards to COVID-19 tests. There are two kinds of tests that are being talked about an awful lot at the moment with regards to COVID-19. The first one is the one that I talked about earlier. It's the PCR testing, and that is a nose or a throat swab. It's a nose and a throat swab, and it's quite an unpleasant swab to have done, and some of you may have had it done. And really, to do it properly, you have to make the person who's having it done really should be gagging. It's unpleasant, it's nasty. You have to have a swab right to the back of your throat, and this tells you if the person has the disease at this point in time. So if I developed symptoms, I'd get a test. It would be the PCR test, and it would tell me either yes, I've got COVID-19, or no, I don't. But there is a problem with false negatives, and we're still learning, but anything up to about 30% of the time the test result might be a false negative. And then antibody testing is the other kind of test, and this is a blood test. So the PCR testing is a nose and throat swab. The antibody testing is a blood test, and this just tells us if somebody has been exposed to COVID-19 in the past. And what you're doing is you're looking for antibodies in the blood. But it's important to remember that just because somebody's got COVID-19 antibodies, it doesn't necessarily mean that they are immune to COVID-19. So from our point of view, the antibody testing is incredibly important because it's telling us how many people have been exposed. It gives us a lot of information, but it doesn't change anything. We're not going to say, well, yes, you can go back to work because you've got a positive antibody test. It doesn't work that way. And I'm not going to say any more about that because Neil is going to be doing a fabulous session on Thursday about immunology, and he will be covering this in more detail. But I did want to just compare and contrast the two different types of tests that we currently have. So, in conclusion, microbiology, and I think if you learn nothing from today, microbiology is much an art as it is a science. We tend to want absolute answers, and it's either positive or negative, or somebody has the disease or they don't, and it's not that simple. And I hope that's something that you've taken away from this session. And of course, we can't get rid of bacteria and viruses. That's impossible. And even if we could, we wouldn't want to. So we need to manage them by this chain of infection and by breaking the chain. And in future modules, we'll be talking about how we can break this chain of infection now that we've learned a bit more about the infectious agents. Testing is incredibly important, but it has its limitations. And as I say, the chain of infection, we need to learn how we can break that. So I want to say thank you all very much. I'm sorry for the IT gremlins and my mother phoning up halfway through the session. I think today is a bit cursed. So if we could stop the recording, Joe, please. And does anybody have any questions?